Welcome back to Psych 351, where we're going to continue on discussing Chapter 8, the control of movement. In this particular lecture, I'm going to move on and talk about the brain's control of movement. Now, for this unit, I'm going to cover just the basics. You're going to see that these circuits look complicated. However, compared to what's really there, these are very simplified diagrams. But I'm going to hit just the highlights, and in terms of what I want you to memorize and know for the test and the final, please focus a lot on what I go over here. There's more detail in the book than I want you to memorize. I want you to read the chapter, but in terms of memorization and what you need to memorize for the exam, what I'm going to go over now is the bulk of it. At the end of this lecture, I want you to be able to explain the basic organization of the motor cortex. I want you to name the brain regions that make up the basal ganglia. I want you to explain the function of the basal ganglia, including both the direct and indirect pathways. Now, hopefully you're familiar with the term somatotopic. We've talked about this with primary somatosensory cortex, where certain parts of your body have a greater concentration of nerves. If you look at the cortex, you'll see that consequently there's a greater amount of cortex devoted to those greater concentrations of sensory neurons. I mentioned before with the alpha motor neurons and the motor unit, some places have a smaller motor unit. That means we're going to need more alpha motor neurons. We're going to end up having more cortex devoted to them. So that's exactly what we have here. Here we've got, instead of a sensory homunculus, we've got a motor homunculus. So if we look at the precentral gyrus, also known as the primary motor cortex, and let's say we stuck an electrode in here and stimulated this area here, we would look to see what moved. And by doing that all across here, we can develop a map. So let's say I put this in here and stimulated here, I would see movement related to the shoulder. If I did that here, I'd see movement related to the palm of the hand. And you get the idea how we can map this whole thing. Each person has a slightly different homunculus. If I were to stimulate this area in one person, we might see movement related to their leg. If I did it in another person, it might be a slightly different part of the leg or the abdomen. Now, one thing that's important to understand when we're talking about the premotor cortex, does not control, that does not equal control of individual muscles. Okay, so when I stimulate here, it is not as though just the deltoid muscle twitches. When we stimulate the primary motor cortex, we get movements. So not individual muscles. That's controlled by the spinal cord and some of those reflexes we talked about before. We get complex movements involving many different muscles. The longer I stimulate, the more complex the movement. So if I stimulate the shoulder here for, say, a second, I might get a little movement. If I do it for 5 seconds, I may get a full rotation of the arm or something like that. Now, other parts of the brain that we're going to talk about don't seem to have quite the same topographic or somatotopic organization as the primary motor cortex. So what I've shown you here applies to primary motor cortex, and that's it. Now, when we want to initiate a movement, ultimately, we're going to stimulate the primary motor cortex, and that is going to send signals through several different pathways down into the spinal cord where the muscles are controlled. Do not worry about memorizing those pathways. I just want you to know that it starts with the primary motor cortex. Now, the primary motor cortex doesn't do it all on its own. It gets a lot of feedback. If we want to plan a movement, we want to engage in a sequence, we need to involve other parts of the brain. So we're going to have lots of connections to that primary motor cortex, lots of inputs. We'll also have lots of outputs. Here is a simplified diagram. This is a good one for you to know. This is a simplified diagram of the cortical control of movement. Remember, obviously the spinal cord is important, but I want you to focus on the cortex when you're studying. Here in our association cortex, we're going to have visual association, auditory, somatosensory association cortex. These are where your perceptions, your memories, all of those things are stored. In order for you to move or decide you want to move, to generate the motivation to move, all of these things are going to come into play. And what those do are send signals to the prefrontal cortex. Okay, so the prefrontal cortex collects all this information. If you're going to move, it's this prefrontal cortex that seems important for generating plans to move. Okay. Once you decide you want to move, now you have to actually engage in that behavior. I want you to know that the prefrontal cortex is going to send information to this pre-supplementary motor area as well as into premotor cortex. Premotor cortex is going to send information onto the primary motor cortex. Now we also have something called the supplementary motor area. That's going to send information on into the primary motor cortex also. So you can see a couple of the big inputs here just from the cortex. Remember, it's just the cerebral cortex. We're going to have the supplementary motor area coming in, and we will have the premotor cortex coming in. Now I'm going to oversimplify and summarize functions a little bit here for you. This supplementary motor area is very important for behavioral sequences. So in this case, let's say the book talks about an experiment where a monkey had to push a lever, then turn the lever left, and then they got a peanut. Okay. Now if they turned left then pushed the lever, if they did it out of sequence, they wouldn't get the reward. So here the sequence is critically important. If they get that sequence incorrect, no reward. If there's damage to this supplemental motor area, they can still move. They can still push a lever. They can still rotate their hand to turn the lever left. But they can't. They have great difficulty doing it in the correct sequence. Okay, so that's how we know what this supplementary motor cortex is important for. Other studies in humans have shown that there's a similar function. So if people have a stroke that involves a supplementary motor area, they'll have difficulty with behavioral sequence. If a person is asked to do a behavioral sequence, they will end up showing activity using fMRI in this supplementary motor area. The premotor cortex is going to be critically important for executing complex movements that are guided by sensory information. And here, what's important to remember is we're going to be talking about arbitrary stimuli. So, what do I mean by arbitrary stimuli? If we have, say, a red light and a green light, and if I flash the red light the animal has to point left to get a reward. If I flash the green light, they have to point right. The red and green don't necessarily indicate left or right. They don't indicate position. Okay, so right in front of them, they'll have a signal that doesn't move. Red light, they have to point left. Green light, they point right. If we lesion the premotor cortex, they'll have difficulty doing that. Now, if I have, say, a red light and I put it to the left, and a green light and I put it to the right, and if the red light flashes, even with damage to the premotor cortex, the animal can point to it. Okay, so it's really in some of these complex learning paradigms where the premotor cortex seems to play an important role. Now, as you know, this primary motor cortex is ultimately going to lead to the movement of muscles by causing the activation of descending spinal pathways. Now, there's a kind of a simplified way of looking at it. This is from a different textbook, but I think this is a great way to think about it. We can really think of 4 different systems being involved in movement. We're going to have these systems here, these descending systems. We'll have motor cortex, where this starts, and then brainstem centers that are important for regulation of postural control. These are both going to be influenced by activity of, say, the basal ganglia and the cerebellum. Okay, so these systems are going to influence activity here. These systems, these descending systems, are then going to send information onto the lower brainstem circuits and the spinal cord. And this is where we're eventually going to get muscular contraction. So we'll have local circuit neurons in the spinal cord. Right? These are going to be involved in reflexes. So I talked about the monosynaptic and the polysynaptic reflex in a previous lecture. Those are the neurons here. We'll also be talking about the alpha motor neurons. Those are sometimes called lower motor neurons. And you know that those innervate the skeletal muscles, so when these get excited, right, we're going to get contraction of the muscle. So these upper motor neurons are going to end up eventually and indirectly causing the contraction of muscles. Their activity is modified by other brain regions, and we'll talk about the basal ganglia in a minute. In turn, they can influence local circuit neurons. So I talked about that inhibition, right? Almost always, this regulation of local circuits is going to be inhibitory from the brain. And I did mention if you sever this, often you will see a lack of inhibition. That's called disinhibition. And we'll see very strong reflexes. And that's a very bad sign. Now, the basal ganglia are exceptionally complex. And I don't want you to know everything there is to know about the basal ganglia. There's a textbook for one of my graduate classes that was 300 pages long. And that was just volume 1 of the basal ganglia. And so we know a tremendous amount about it. It's hugely important. It's incredibly complex. I want you to know the parts of the brain that are involved in it. So I want you to know caudate and putamen. So you need to know the name. I want you to know globus pallidus. We're not going to differentiate between external and internal. You don't have to do that for this class. But I want you to know globus pallidus is part of the basal ganglia. I want you to know subthalamic nucleus. Okay? And I want you to know thalamus. Don't worry about VA, VL. Just thalamus is good enough for now. We've talked about the substantia nigra before in previous units. This has a tremendous amount of dopamine. Okay, so the dopamine producing neurons are located in the substantia nigra. They send their axons forward through the median forebrain bundle, the MFB. I hope that's a review. Okay. Now, the caudate and the putamen are sometimes called the striatum. Okay. So here we've got the substantia nigra sending axons up to the striatum. Okay. I hope you now recognize this as our nigrostriatal dopamine system. If that's not review, study up on the nigrostriatal dopamine system. I'm going to be talking about it over and over throughout this course. It's hugely important for many things, from movement here to Parkinson's disease when we talk about the neurological neurodegenerative disorders to addiction. All of these things involve the nigrostriatal dopamine system. All right. So you do need to memorize those brain regions. You don't need to worry about the connections too much. I'll let you know specifically where I do want you to know connections. We'll focus a little bit more on function. So I want you to know two things. We're going to make a distinction between a direct and indirect pathway. Okay? Now, basically, the basal ganglia form part of a loop, a feedback loop. So you can see we're going to go from primary motor cortex. We'll send information into the basal ganglia through several different ways. That's going to basically feed back on premotor cortex, on SMA, and on the primary motor cortex. This feedback is going to help guide the action of these things. Okay? So these don't cause movement per se. There are some pathways, of course, that are going to go down to the brain stem, okay, the ventromedial, one of the descending systems. But most, the primary cortex is going to have other projections to the descending pathways. Those aren't shown here, believe it or not, to try to keep this simple. Okay, so when we talk about primary motor cortex causing movement, I want you to realize that that's not shown in this figure. All right, so the direct pathway. I'm going to walk you through this. Black equals excitatory signals. Red equals inhibitory signals. Okay, so let's follow this direct pathway. I'm going to go from primary motor cortex to the caudate and the putamen. Right now we're going to focus on this direct pathway. I get excitation. I get excitation of inhibition. What does that mean? This is going to be a GABA neuron. So when this GABA neuron's excited, what do I get? The release of GABA, which does what here? It's going to inhibit the globus pallidus. When I inhibit the globus pallidus, what does that do? Well, that means this inhibitory connection is actually going to be inhibited. Okay, so I get less GABA being released. If I get less GABA being released, that's something called disinhibition. If I get disinhibition of this, what does that do? That means now this is going to be excited. When this is excited now, I'm going to stimulate all the motor cortices, and I get an increase in movement. Okay, so by exciting inhibition of inhibition, I get excitation. So the direct pathway equals increased movement. Okay, so this is the bottom line that I want you to remember. Stimulation of the direct pathway leads to an increase in movement. Now let's look at the indirect pathway. That is going to be indicated by these dashed lines. So the motor cortex, again, is going to send signals down. We're going to get excitation of inhibition. So what does that mean? That means I'm going to have more GABA being released. Okay, so we have excitation here. That means I have more GABA. If I have more GABA here, what does that do to this? Right, it's going to inhibit this neuron. Okay, so when this neuron is inhibited, what does that mean? That means less GABA. And I'm going to have disinhibition here. That means this pathway is going to be excited, and I'm going to release excitatory neurotransmitter. Now, when I excite the globus pallidus internal, remember these are inhibitory GABA neurons. So what's going to happen here? I've increased GABA. What's that going to do? It's going to decrease activity. Okay, so if I reduce activity in these pathways, that's going to reduce movement. Okay, so indirect equals decreased movement. So if I give you this information on an exam, you need to be able to figure out what's going on. So you don't need to memorize it, but you do need to understand it. You need to be able to follow it along. It's easiest if you just kind of draw it out and talk yourself through it, I think. All right. So we have direct, which facilitates movement. We have indirect, which inhibits movement. So when you're moving, you're going to actually have activity in both the direct and indirect pathways. So we're going to facilitate movement, but we're going to also inhibit it. And so it's kind of a weird thing to try to think about. To make a long story short, what the basal ganglia do is facilitate desired movements. Okay, facilitate desired movements, but suppressing undesired movements. Okay. Now, one way you can see this is in Parkinson's disease. For right now, don't panic about specifics about Parkinson's disease. We're going to talk a bit more about this later on. But in Parkinson's disease, the substantia nigra, all the neurons in there die. Not all. Almost all of them die, though. About 90% when you start to see symptoms. That means we don't have those dopamine inputs anymore. You can see some are excitatory, some are inhibitory. Okay, so when we get rid of dopamine, that means we're going to affect both the direct and indirect pathways. Okay, so that means we're going to have altered facilitatory and inhibitory effects. And if you think about the symptoms of Parkinson's disease, they're almost contradictory in a way. In Parkinson's, a patient has great difficulty initiating movement. They don't move very well. But at the same time, if you watch them, they have a tremor. They kind of shake. How can we have too little movement and at the same time too much movement? And that is going to be because of dopamine's effect on the direct and indirect pathways. It affects both of them. All right, that's the basics now of the basal ganglia. You got their function, direct, indirect. For this, don't worry about the hyperdirect. We're not going to talk about that. So I hope you can explain the topic, organization of the motor cortex. You can name the regions that make up the basal ganglia. You understand the functions of the basal ganglia, including the direct and indirect pathway. You should also understand when I'm talking about premotor cortex, sort of the basic functions I gave you earlier on. All right, I hope that was as fun for you as it was for me. And we are now done with the brain's control of movement. I'll see you next time.